In Module 3, you learned all about the structure of cells. In Module 4, we are learning about how cells function. This is called cell physiology. And this has to do, again, with the Stirringer life processes, which, as you have learned, take place at the cellular level. So today we're going to be looking particularly at cell transport, which is the T in Stirringer. If you could please make sure you have out your graphic organizer for Module 4, which can be found at the end of Module 4. It's two-sided. We're going to be starting with the cell transport side, which, as I mentioned, is a Stirringer life process. Transport is the T in Stirringer. Transport is a process that lets food in for nutrition. There's a place on your graphic organizer to write that in. Cell transport also lets oxygen in for respiration. There is a place on your graphic organizer to write that in. And cell transport lets waste out for excretion. There is a place to write that in as well. So as you can see, transport is related to several other Stirringer life processes. N for nutrition, R respiration, and E excretion. Cell transport takes place through the cell membrane. Materials are able to move in and out of cells through the cell membrane. There's nowhere to write that on your graphic organizer, but there is a picture of the cell membrane, which you are going to label. There's also a notation where you need to fill in a blank. Materials move in and out of cells through the cell membrane, and then there's a two-word space. It is a selectively permeable membrane. So please write those two words in the blank spaces. Selectively permeable means that the membrane allows certain materials through while preventing others from going through. If you look over on the right upper corner of your graphic organizer, there is a diagram of the cell membrane. Notice that you are looking at a greatly enlarged picture of the cell membrane, where on the outside of the little dots at the top, you are on the outside of the cell, and on the inside at the little dots at the bottom, you are on the inside of the cell. This picture doesn't show you a cell at all. It just shows you an enlargement of the membrane that surrounds the cell. Please label the large molecule that is, the label is at the bottom left corner, a membrane protein. Proteins are one component of the cell membrane. But the membrane is largely made up of these smaller little dot-looking molecules. If you look at them closely, you will see that each dot actually has a head and a tail. And there are two layers of those dots that meet in the middle. This is termed a phospholipid bilayer. And there's a place to write that in as well. So these are types of lipids, which you studied back in Module 2. You will recall that lipids are major components of the cell membrane. It's one of the most important functions of lipids. It's a bilayer because there are two layers of these lipids that come together. In just a couple of minutes, we're going to take some notes on cell transport, but before we do that, we're going to look at a demonstration to illustrate the process. So here we have a picture of an experimental setup where there is a plastic dialysis bag placed inside of a cup. If you look at the picture carefully, you will see this dialysis bag. The dialysis bag is made of plastic dialysis tubing, which is used in kidney dialysis. You may have heard of that. It's similar to the cell membrane in that it is selectively permeable and lets only certain materials in and out. Inside that dialysis bag is a starch solution. Starch, you will remember from Module 2, is a long chain of monosaccharides. So it's a very large molecule While on the other hand, on the outside of the dialysis bag is an iodine solution. That's why the water looks like a yellow orangish color. Iodine is a very small molecule. Over time, if we allow this dialysis bag to sit in the iodine solution, the iodine molecules, because they are very small, passed through the dialysis membrane. They diffused into the bag. This is cell transport. This is transport, similar to what happens in a cell. And we have evidence that this happened because as the iodine came into contact with the starch solution turned colors. You learned in Module 2 that this is an indicator for starch, and that is what happens in the presence of starch. We also have evidence that the starch molecules did not move out of the bag. They stayed in the bag because they were too big to pass through the dialysis membrane, since it was selectively permeable. The solution in the cup actually stayed the yellow-orange color. So once again, in this demonstration, the dialysis bag represents a cell, and the molecules moving through the membrane, iodine, represents cell transport. There are a couple of different types of cell transport that we're going to take a look at. It's really important that you listen to the explanation of the different types of cell transport rather than just quickly trying to record what's on the slide. So let's start by looking at passive cell transport. There is a place on your graphic organizer to write in this information, but please write it in as we talk about it. So first, on the side for passive cell transport, you need to record that this is the type of transport that does not require energy by the cell. The reason that it does not require the input of energy by the cell is that it is moving down the concentration gradient. If you look at the picture, the illustration, it very clearly shows you how this is the case. So on one side of the cell membrane, you have a fair number of molecules, a high concentration. While on the other side of the cell membrane, you have just a few molecules, a low concentration. So you are moving from a high concentration to a low concentration, which does not require energy input by the cell. Think of it as sitting at the top of the hill on a bicycle. You can coast down the hill without pedaling because you are at the top of the hill. Moving from high to low does not require any energy. You are going down the gradient. The gradient is the hill. On the other hand, active transport does require the use of energy by the cell because it is movement against the concentration gradient. Notice in the picture, look particularly at the orange triangular or diamond-shaped molecules that are moving across the cell membrane. On the outside of the cell, there are only a couple. On the inside of the cell, there are several. It's moving from a low to a high concentration, and in order for this to happen, it has to go against the gradient. It has to go up the hill in a sense. If you are at a bottom of the hill with your bicycle, you're going to have to pedal to get up that hill, to go from a low to a high level, requiring the use of energy. Same thing happens in cell transport. On your graphic organizer, if you could look down at the section titled Diffusion, you'll notice that there is an arrow leading from the section on passive transport to the section on diffusion, because diffusion is a type of passive transport. The first thing you need to do is define the term diffusion in the space provided. Please define that term as movement of molecules from a high to a low concentration. Once you've done that definition, you need to have a look at the example that is shown both on your paper and here. Let's make sure you understand what you're looking at. You are looking at an alveolus, which is a part of your lung. where the oxygen comes in, and that alveolus is surrounded by a blood capillary. So when you breathe in, your lungs fill with oxygen, and you can see that oxygen in the alveolus, and it's in a high concentration. The blood is going to take that oxygen. It will move to a low concentration in the blood so that it can be delivered to the cells of your body. Meanwhile, in the cells of your body, carbon dioxide is being produced. The blood picks it up and carries it back to the lungs. So CO2 is in a high concentration in the blood. It's going to move into a low concentration in the lungs so that you can exhale that waste product back out. This is going to happen until molecules reach equilibrium. If you will look on the far right side of your graphic organizer, you will see the note that molecules move until they reach equilibrium. You need to define the term equilibrium in that box. An equal concentration on both sides of the membrane. Please make sure that you understand the diagram and that you understand that oxygen is moving from a high concentration to a low concentration, and carbon dioxide is moving from a high concentration to a low concentration. Now if you'll take a look at the box labeled osmosis. Once again, osmosis is a type of passive cell transport, so you'll see an arrow leading from the section on passive cell transport to the box on osmosis. Please make sure that you define the term osmosis in the place provided. It's defined as the movement of water molecules from a high concentration to a low concentration. It is a type of diffusion, but it specifically refers to the movement of water. You have an example both on your graphic organizer as well as on this slide. You can see that in normal circumstances, red blood cells look similar to a donut. Those are the cells up at the top. But if you place these cells in a distilled water solution, it's very concentrated water, it is a high concentration of water, the water will move into the cells. The water will move from a high concentration outside the cells to a low concentration inside the cells. And in the case of red blood cells, which are animal cells, they can swell up and the membrane can burst. Similarly, if you place the cells in a salt water solution, there's actually less H2O outside the cells. So it's going to move from a high concentration in the red blood cells to a lower concentration in the salt solution. That means water is going to leave the cells and the cells are going to shrink or shrivel. Once again, this happens until equilibrium is reached, an equal concentration on both sides of the membrane. Another Stirringer process that has to do with cell physiology are the processes of photosynthesis and respiration. Synthesis is the S in Stirringer. Respiration is one of the R's in Stirringer. So photosynthesis and cell respiration are both Stirringer processes. If you look at the far right side in the upper corner of your graphic organizer, you will see how these two processes work together in a cycle. Go ahead and write that note in the box. The two processes work together in a cycle. At the top, you will see that plant cells are taking in sunlight. They are producing glucose, sugar, and oxygen. This is used in the mitochondria of cells to produce energy in the form of ATP. As a waste product, those cells are producing water and carbon dioxide, which again is used by the plant cells for photosynthesis. So you need the two processes to work together. Now looking up at the photosynthesis box, you need to start with a definition of photosynthesis. This definition is already partly written for you. You need to fill in the blank. Photosynthesis involves making food, also called sugar or glucose, for nutrition. The equation for photosynthesis is also written out for you. It is absolutely essential that you memorize this equation. If you do not know what the chemical symbols stand for, you should write them in. There is a place for you to do this. It says, write the equation in words. So here is the equation in words. Carbon dioxide plus water plus sun yields glucose plus oxygen. On the left side of the equation, we have the reactants. There is a bracketed area that you need to label reactants. These are the things that are going into the plant cell in order to allow the process to happen. On the right side of the equation, we have the products. There is a bracketed area on your graphic organizer that you need to label products. This is what is produced following the reaction of photosynthesis. Down in the bottom section of that box, you will see that it asks you which organelle is this occurring in. The organelle is pictured there for you. This organelle is the chloroplast. So this process only occurs in plant cells because those are the cells that contain chloroplasts. These cells also have the green pigment chlorophyll, which capture light energy. Write that in on your graphic organizer beneath the section where you are asked for the organelle. It's also important for you to realize the reason that plants are green. Green light is the wavelength that is reflected by the plant. All of the other wavelengths of light, from red, and blue, orange, and white, all of those wavelengths are absorbed. Green is reflected, giving them their green color. Now let's look at photosynthesis in terms of what is going in to the plant and what is coming out of the plant. We can find this out by simply looking at our equation. We have three things going in, and you will see that there are three arrows pointing toward the leaf. These are the three arrows at the bottom. You should label each of these arrows. The first one, CO2. Carbon dioxide is going in. The second arrow, H2O. Water is going in. The third arrow, sun. Sun is going in. We can also look at the equation to determine what is leaving the plant, what is produced by the plant. You have two arrows pointing outwards. You should label each of those arrows as well. The first arrow, C6H12O6, which is glucose being produced by the plant. The second arrow, O2, oxygen being produced by the plant. As I previously mentioned, the processes of photosynthesis and cellular respiration are going to work together in a cycle. Here is how. This is already written on your graphic organizer in big, bold print. The food that is made in photosynthesis, that's the glucose, is used to make energy for cells. This energy is in the form of ATP, adenosine triphosphate. Food is not energy. Food contains chemical energy, but living organisms must convert that chemical energy to a form that they can use. That form is ATP. If you look just beneath the cycle in the upper right-hand corner of your graphic organizer, you will see the same diagram that is on this slide. A diagram showing you the structure of adenosine triphosphate. The 3 P's represent the 3 phosphates. Adenosine triphosphate stores energy in a chemical bond. The chemical bond is what holds the phosphate groups together. When one of those bonds is broken, energy is released. That's why there is an arrow showing the release of energy between the second and third phosphate group. In the space on your graphic organizer, just beneath that burst of energy, you need to write in ATP is the energy molecule for cells. Somewhere else on your graphic organizer, there is a bit of space to the left of the energy burst. You need to make a note that energy is stored in a chemical bond, and when the bond is broken, the energy is released. Now we're going to look at how cells use the food made in photosynthesis to make energy in the process of cellular respiration. The definition is already partly written for you, but you need to fill in the blank. Respiration involves using food made in photosynthesis for respiration to make ATP for energy. The equation is again written for you, and it is essential that you memorize it. But pay close attention to the equation. It is essentially the opposite of photosynthesis, with just a couple of changes. Therefore, if you have memorized the equation for photosynthesis, you should also be able to memorize the equation for cellular respiration by simply reversing them. The products of one are the reactants of the other. Let's write out this equation in words. You have a space to record this on your graphic organizer. Glucose plus oxygen yields carbon dioxide plus water plus ATP. The differences between cellular respiration and photosynthesis are that there is no sunlight necessary for cellular respiration, and that ATP is one of the products. You also need to label on your equations in the bracketed area the reactants on the left side, that's what's going in, and the products on the right side, that's what's coming out. The organelle where this is occurring, and again there is a space to record this, is the mitochondria. And mitochondria is found in every living cell, plant and animal. So both plants and animals carry out the process of cellular respiration. Let's label on the diagram of the mitochondria what is going in and what is coming out. You will see 2 arrows going into the mitochondria. Looking up at your equation, you will see that glucose, C6H12O6, and oxygen, O2, are going in. So label the 2 arrows that are going in. You also need to record what is coming out of the mitochondria. There are 3 arrows. Looking at your equation, you are going to label them CO2, carbon dioxide, H2O, water, and ATP, adenosine triphosphate, the energy molecule. Now remember that cellular respiration can take place with oxygen or in some cases without oxygen. There's quite a bit of information that you need to record here over in the right bottom corner of your graphic organizer. With oxygen is called aerobic respiration. And the benefit to aerobic respiration is that it makes a lot of ATP for energy. So in the box that says with oxygen, you need to record both of those things. Aerobic respiration makes a lot of ATP. There is a lot to write in the box for without oxygen, so write small. In the box for without oxygen, the term for that is anaerobic respiration. But there is another term that is often used. So not only do you need to write anaerobic respiration, but you need to write the term fermentation as well. Now, if you will follow the 2 arrows on the far right and the far left of that box on the slide, there are some differences in fermentation depending on the organism. You need to record these differences. In plants and in microorganisms like bacteria, fermentation takes the form of alcoholic fermentation. This is how grapes produce wine. This is how yeast produces beer. In animals, on the other hand, fermentation takes place in the form of lactic acid fermentation, which is what makes your muscles burn when you've been exercising for a while and you are having trouble getting enough oxygen to those cells. Make sure you have recorded again anaerobic respiration, also called fermentation. In plants and microorganisms, it's alcoholic fermentation. In animals, it's lactic acid fermentation. And finally, you need to record that this process only makes a little ATP.